Till now we had seen membrane fusion process. So how it was happening? Viral membrane fusion involves lipid membranes coming together. GP41 fusion molecule plays critical role in membrane fusion, enables the virus to penetrate the cell membrane, allows the lipid layers to merge and virus to enter the cell. Receptor blocking approach. CD4 molecules are there. They are the target. So developing drugs to block the CD4 receptor prevents the virus from entering the cells, and current challenges include the potential toxicity to normal cells. Viral entry receptors and co-receptor we had seen. AIDS development. It is not immediate upon viral infection. Average time for infection to full AIDS requires 7 to 8 years. Requires progression through multiple stages, like involves immunosuppression, opportunistic infection, secondary neoplasia, and neurological dysfunction. Infection characteristics are: initial infection primarily targets the macrophages and hence uses the CCR5 co-receptor. In advanced stages, the virus mutates and develops the ability to target the CXCR4 receptors and becomes more aggressive towards the helper T cells. So initially, macrophages are the target, and later the T helper cells are the target. So HIV virus initial interaction, receptor binding with the GP120, and then with CXCR4 helper T cell infection, and with CCR5 receptor macrophage infection. And when there is viral mutation, it will infect helper T cell. Next thing is HIV infection and disease progression. Initially, it was believed that HIV infection हुआ तो वो inevitably it will lead to AIDS. But surprisingly, we saw that some individuals resist AIDS development. In genetic variations, this is a very important thing. So not the European descendants. They have mutations in the CCR5 gene. Population distribution: 1% completely they are resistant to AIDS, and 10 to 15% are partially resistant. The homozygous mutations, complete resistance. The ones who show complete resistance, both maternal and paternal genes are defective. Natural immunity: virus cannot effectively infect the cells. So represents the approximate, and that represents 1% of the population. So if someone has homozygosity for this particular gene, for example AA, then it will show complete resistance, and the virus won't be able to infect the cell. And this particular homozygosity, it is for CCR5 receptor. So if CCR5 is defective, then if this particular RCCR5 is defective, then what will happen? Even if this interaction occurs, but this interaction will be problematic and the virus cannot infect the cell. And when there is heterozygous mutation, this type of mutation is in the gene which will make this particular receptor, then so one gene is normal and other gene is defective. Virus entry will take place, but multiplication significantly slowed. This virus will enter the cell, but its multiplication will be slow. Viral entry mechanism: virus interaction with GP120 protein we saw. So CCR5 receptor activation. In normal case, efficient viral entry, but in case of mutation, reduced or blocked viral entry. Viral entry, we saw this. Pharmacological intervention here: CCR5 receptor blocking. And the drug here is the Maraviroc, also spelled Maraviroc. Okay, what it will do, it will block the CCR5 molecule interaction. This interaction it will block, and it will prevent the GP120 from binding and entering the cell. Okay, so CCR5, it is a chemokine receptor crucial for HIV cell entry. If it is homozygous, both genes copies mutated. If heterozygous, one gene copy is mutated. GP120 is the viral protein facilitating the cell entry. Drug molecules that block the CCR5 receptor we saw, and hence virus binding and entry is prevented. And this particular drug, it is positioned between virus and the receptor. This drug, it is positioned between virus and the receptor. Okay. GP120 characteristics काय सांगता है? Constantly changing, it is a constantly changing molecular structure, described as playboy protein with variable appearances. So protein sequence determined by viral gene, and if the gene that goes rapid mutation, then amino acid sequence will constantly alter the virus. So if initially the glycoprotein is like this, and if there is mutation, how will it look? It will look something like this. Another mutation will make it look something like this. Okay, so these are the mutations. Mutation challenges: mutation impact. Frequent genetic changes makes the antibody recognition difficult. So here problem is with the antibody. Whenever there is an antigen coming, our body forms antibodies. But if the antigen keeps changing, then the antibody recognition is difficult. So gene mutations cause protein structure changes and prevents consistent immune system response. Hypervariable region, we saw this. Specific mutation prone area on GP120 rapidly mutates and allows viral evasion of immune response. Immune system challenge: antibodies become ineffective quickly, and because of this, constant molecular transformation, vaccine development obstacles are there. So no successful HIV vaccine is there, because genetic variation of GP120. What are the research challenges? Constant molecular changes are occurring and difficulty in creating stable immunological target. So HIV virus, primary attachment with GP120 and CD4 receptor, then interaction points: CCR5 receptor in the macrophages, CXCR4 receptor in the T helper cell. This particular GP120, it has hypervariable region, it does frequent mutations. ठीक है? Now what happens, that neutralizing antibodies that bind to the GP120 and prevent the viral cell attachment and become ineffective with viral mutation. We know that antibodies become ineffective due to rapid viral mutation and emergence of new GP120 variants are there. So there is viral neutralization process by antibody. These neutralizing antibodies, they bind to the GP120 and prevent this viral cell attachment. So this is our GP120, and if against this an antibody has formed, it will prevent its attachment to the host cell. But this particular thing, these neutralizing antibodies, they will become ineffective when there is huge viral mutation. Continuous monitoring of GP120 variants, understanding their mutation patterns, and developing adaptive therapeutic approaches is how tracking a viral evolution is occurring. So HIV virus, GP120 protein receptor binding to CD4 receptor, co-receptor interaction, then CCR5 or CXCR4, then cell entry because of GP41 activated viral cell fusion and viral infection. Next thing, what is it? HIV virus demonstrates remarkable adaptability, constantly altering AS despite maintaining core attachment mechanism. Even though these genetic changes, they are changing the antigenic structure, but the virus, it will maintain its core attachment mechanism. So virus strategically changes its space to evade the immune system recognition, but it keeps its core attachment. That means this is our GP120, this is our host cell, this is our GP41, and this is our double membrane wala lipid layer, double layered lipid, bilayer lipid. So this is the GP120 changes its spaces, it makes its spaces weird, but it keeps its viral entry वाला जो mechanism है ना, वो तो ये same रखता है. That means how the immune system will recognize these viruses, it is confusing that, but how it will enter the viral cell, that remains the same. Core attachment mechanism, it remains the same. It is basically only confusing the viral, the antibodies formed against virus. Viral protein structure we have known, this also we know. Inhibition strategies, attachment inhibitors block initial receptor binding, fusion inhibitors prevent membrane fusion after attachment. Drugs targeting GP41 can tie up the viral proteins and prevent the cellular invasion. Vaccine developing challenges we have known. Drugs, what will they do? They will either prevent the viral attachment, block the membrane fusion, or interrupt the viral replication cycle inside the host cell. So attachment inhibitor, CCR5 receptor is the target, and which drug we use? We use Meraviroc. We use which drug? Meraviroc we use, and it blocks the initial viral binding. And fusion inhibitor that we use, that targets the GP41, it does not let the membrane fuse. And which drug is here? Enfurovertide. Enfurovertide is the drug. Maraviroc.